So welcome to our first NU-HTA user spotlight. So we're just trying to get the word out about NU-HTA and show what great work our users are doing. So for those of you that don't know, Northwestern, we're the high-throughput analysis lab. We're a core facility on the Evanston campus, but we are available to both campuses. Anybody that wants to use us can come down and visit. And we do laboratory automation. We provide laboratory automation and tools for drug discovery. And you can contact me. I'm Sarah Fernandez-Dunn if you're interested in talking about what we can do. So for the seminar today, Monica Rosas has kindly agreed to be our first speaker for this series. So she is in the Center for Structural Genomics and of Infectious Diseases. And so she's going to give us a talk today about a SARS-CoV-2 protein that she's working on. Thank you.
Well, hello everyone, and thanks for being in person today and online. And thanks to Sarah for inviting me to present our latest paper. And I'm really happy to be here and talk to you about our work where we show how manganese coordinates the Cap0 RNA to align substrates for efficient transfer of 2' methyl transfer by SARS-CoV-2 and Asp16. As Sarah mentioned, this work was done at the Center for Structural Genomics of Infectious Diseases at Feinberg School of Medicine in Northwestern University. Just a brief reminder that coronaviruses are all over the place. It's just that we have this named low pathogenic coronaviruses, which causes the common cold, and also these highly pathogenic coronaviruses. The highly pathogenic coronaviruses are very dangerous, and if you have noticed, one of them is the SARS-CoV-2, which is the agent of the COVID-19 pandemics. And all of these coronaviruses, they are zoonotic. It means that they jump from the reservoir to another animal intermediate and then from them to the humans. They are very concerning because we've already registered 3 of them, more of this kind of zoonotic diseases can come in the future, and we should be ready for that. Then now we have vaccines, and that's really good. So we are able to prevent most of the infections. However, we also have new variants. And then what's next? Well, we need to check for people who cannot get vaccinated because several reasons or health conditions. And then maybe some of the vaccines will need some boosters, maybe we'll need more vaccines, maybe we will need some drugs or combined therapy. Then that's why many people keep working all over the world to find new ways, or at least to find new targets that can stop the viral replication. And one of the great things that happened during this year is that several efforts described the life cycle of the virus since the entry, how the virus gets into the cell, it releases its genomic material, then how they get translated, all these proteins later on, the importance of these proteins of being cleaved to form all the replication machinery. Then thanks to this, we now can use some of these nucleoside analogs that have been somehow useful for treatment, but there is a lot of research also trying to block the entry or some of the proteases that will release these polyproteins to form this complex. And one interesting process that happens in the virus, and that is very important to avoid the immune response in the host, is the capping. And is this modification in the 5', yes, now you can see it, the 5' position of the RNA that needs to be modified in order to avoid the recognition by the cell, and then also improve the translation, which will be really helpful to target if we can avoid this step. And then it is known by previous studies from SARS-1 that blocking the capping actually is a good target that activates the immune response in vitro and in vivo studies. So that's why we decided to take a look on this process. And then the capping was previously described for SARS-1 and other coronaviruses. And it is known that it starts by cleaving the first phosphate of the nascent RNA. And that process is performed by the non-structural protein NSP13. Then the next step is transfer AJMP from a molecule of GTP to this cleaved RNA, and then this process was recently described to be done by NSP12. Then the next step is the transfer of a methyl group that comes from S-adenosylmethionine to the guanosine group, and that process is performed by the complex NSP10 and NSP14. Then the latest step of this process is the cap in the 2' position, and then the substrate of this reaction is the cap 0, where we only have the methyl group in the position 7 of the guanosine. And then we need magnesium, manganese, and also SAM. I don't know what happened here, but we need S-adenosylmethionine, magnesium, or manganese. And then this process is performed by the complex NSP10, NSP16, which is the star of today's talk. The NSP16 is the catalytic subunit, and it is very important to say also that it is inactive in the absence of NSP10. And that's how finally we get the product, which is also named CAP1, that has these two modified, which has these two methyl groups in the RNA. Then we took a look on what was previously done for coronaviruses in general, and we realized that actually NSP16 is 93% identical to SARS-CoV and 66% identical to MERS. NSP10 is very well conserved, also is 99% identical to SARS and 59% identical to MERS. Then all the job that was previously done for these two coronaviruses was really useful to find some possible druggable sites in this complex, and also to try to improve what we could do from the structural point of view to find druggable sites. So what we saw is that the cap binding site was semi-conserved, that the SAM binding site was really conserved, that the interface was really conserved, which is really good because it was proved to be a good target too. But what really we didn't knew, and it was no information about, is the RNA binding group or the RNA binding site. Then we also checked the previous structures determined for SARS and MERS, and we realized that although it was a lot of work done before, it was still a lot of unknowns. And finally, if you take a look on the PDB, you will see that for SARS-1, there is only a couple of structures, only with one of the substrates. For MERS, we have more information, actually. We have structures with a cap analog, SAM, which is the substrate, SAC, that is the product, cenefungin, which is a very good inhibitor, but a very bad drug. And for SARS-CoV, we have COVID-2, we have more structures with a little bit more information. And also, one of the structures that we determined in the center was this structure that I am showing here, where we have this cap analog. And it means that we have only the first nucleotide of the RNA, together with the substrate, S-adenosylmethionine, and also some sulfates. And that was really good because that gave us a hint on how the RNA could accommodate, because the sulfates mimics the stereochemistry of the phosphates been bound to the proteins. So that was really good, but still we didn't knew how the metal or the RNA accommodates in the protein, and specifically the role on the catalysis. Also, another really important issue is that many people start building models of this structure of SARS-CoV-2 based on the structure of dengue virus. And dengue virus is somehow different. The RNA binding group is deeper, and the positive charge is like more intense in this groove where the RNA binds. And if you take this model, actually it will give you a wrong accommodation of the RNA, because if you see here, like the blue is not super intense and well marked. And also, I don't want to overwhelm you with structural information, but in the past for SARS-1, they were modeling like the first nucleotide, even like just around here, and it will clash. So there is a lot of misinformation about it. So we decided to take over and really try to find the best way to generate structural information for drug design. So we decided to take over a very basic science questions and solve them. So first, what's the role of the metal in the catalysis? So we know that the metal is very important for the catalysis, but where is it binding? What is the metal doing? And how it interacts with the protein, that was unknown. So we'll try to understand these questions or answer these questions, and also how the cap RNA binds to the protein. Then we already had a nice setup for the pipeline. So this is the CSGID pipeline, where we get the protein production, purification, and after purification, it goes either to enzymatic activity, or we just set up high throughput and crystallization. We already knew the conditions and the best screens to get high quality crystals. So we got these crystals, and then in order to know and understand the role of the substrates, metal and RNA, then we performed soakings. And this job is mostly done by Ludmila Shuvalova, associate professor, so she takes over from crystallization to all this great job. And then once we have those crystals, then they are sent to Argonne National Labs, where Joe Runsell usually helps us to collect data, and also George Nasov collects data, and then between the two of them, they determine all the structures that we've working with. And then that's me. I usually take care about the protein production, purifications, then some crystallizations, and the enzymatic activity. And one of the things that I like to highlight here is that it was a very good reason why people could not ever get any RNA bound to the protein, and that's because many of them, they were working with very long RNAs. So last year, it came out this paper describing the structure of the 5' UTR of the RNA. And then we saw that there is like this small linear portion of the 5' UTR that had like 6 nucleotides. And we thought that this could be really good substrate based on our structure with the sulfate bound, which is like at least 4 or 5 nucleotides can accommodate. So we decided to take this short RNA, and then the issue with short RNAs is that you cannot make them enzymatically. They should be chemically synthesized. So we found a company who synthesized that for us, and that's how we get into these experiments. So again, I really like to highlight that the reason why we got this structure is because we did a ton of experiments, or Luda did actually, a lot of soakings, testing different incubation times using manganese or magnesium, the SAM and the cap RNA. Then freeze the crystals and send them to Argonne National Lab to collect data and get the structure. So the first time that Luda told me George got the structure with RNA bound, I was really happy. And then he said, oh, and we have also the metal bound, and I was even more happy. And I'm showing you here like the first structure. So we have all the RNA, it's not all the RNA, but we can see at least the first 3 nucleotides, how they accommodate, and also clearly the metal bound. And this is the structure with the RNA and the substrates. So this is cap 0, you will see here. This is cap 0, it doesn't have the methyl group. But then that was not it. The next time when Luda told me and George told me, oh, you know what? We have the cap 1. Then at that time, I was just like running in circles and screaming because it was really nice to see that the reaction actually happened during soakings in the crystal. So you have a substrate, you have um, both substrates, then now you have the product. As you can see here, S-adenosyl homocysteine doesn't have any more the methyl group, and now the 2' position of the ribose in the first nucleotide has methyl group. So that was really nice. I was really happy about it. And the second thing that was really interesting was that this just happened in the presence of manganese. So the other one was magnesium, and then manganese. And I was like, wow. We know that both metals, they catalyze the reaction. So that gave us a hint, well, maybe there is like a preference for manganese as NSP14, for example. NSP14 only uses manganese and magnesium doesn't help. But then we said, well, maybe manganese is very important, right? So we need to go back and check the activity and see how this happens in reaction and really test the importance of the metal. So we built the team activity. And then it was, by that time, it was still the pandemics and the lockdown. So we were Carla, Paul Hoover, and me literally thinking how to set up this assay. Because, you know, methyl groups, they are not easy to measure. This transfer doesn't generate any color or any redox reaction that you can couple to something else. And all the previous publications for SARS-1 or MERS, they use radioactive isotopes. So we were really thinking about it, and that's why Paul was really helpful on that, because he can handle radioactivity. We set up, and then we saw that it was available, this kit, MTase Glow from Promega, and that we could really adapt it to our assays. And that's what we did. So this assay is based on the detection of S-adenosyl homocysteine, which is the product of the reaction, and then it gets cleaved, produces ADP, and then by luciferase reaction, it produces light, and you can measure the luminescence produced. Then we tested the activity in the presence of different metals and without metals, and also we used another substrate, which is only the cap analog. So it is known that you need like a specific length of the RNA to really get this reaction to happen. So we tested both. And also we were interested in, like from the structural point of view, and translate how important is the length of the RNA. So what we saw here is that only the cap is a very bad substrate. We know it binds because we got the structures, we did ITC, but it's not a good substrate. And we only saw really, really low activity in the presence of manganese. Again, manganese and only manganese. Okay. Then when we tested the RNA, we saw that the highest activity was detected in the presence of magnesium and manganese, which was great. And again, a little bit higher in manganese. It was not the case for sodium or calcium or without metal. So that was really suggesting that manganese is very important. Then when we compared and overlaid the structures of the structure with RNA and only the cap analog, we saw that the second nucleotide here, the U2, is very important to accommodate the first one. It kind of pushes up the first nucleotide and put in place, I will just mention that around here, they are the catalytic residues. And there is a cavity here. So it's the substrate is adenosyl methionine. You can see how it aligns the methyl group to the 2' position. So the reaction can happen. And then if you check the position of the metal, it looks like the metal is actually holding like the RNA in this position, plus this uracil is pushing up the adenosine, and altogether seems to be very important to keep this 2' position in place and get the reaction to happen. Then we also observed the presence of this named promontory region previously by the people who described the first structure of SARS-1. And they kind of mentioned that this aspartic residue was very important, it was very important, but actually nobody knew what it was the real importance of this aspartic residue. And we just realized that it is really important because somehow it's stabilizing the interaction with this second nucleotide, and this second nucleotide is pushing and accommodating the RNA in this position. So that's why our paper says that the metal helps to accommodate the RNA in the right position, and that's how you can improve the catalysis. If you don't have this residue here, as in the cap analog, you can't keep like a stable interaction. You don't have metal, and then the reaction just doesn't happen, or it happens like very slowly, and we cannot even detect it, as in the case of, as you can see here, when we don't have any metal. So that was really good about the metals. So we had the structure and activity evidence about the role of the manganese in the activity. But we also were interested in these aspartic residues, like why we have this aspartic residue and why is it so important. Then we compared, there is only now 4, but in the past there were only 3 structures of RNA methyltransferase in the PDB, and none of them had any metal besides the dengue virus that I showed you previously. And then we checked them. You can see that they actually have different accommodations, and most of the structures, they have like very deep pockets, and the position of the cap is just totally different. We have here like a nice cap accommodated in a specific position with this lock, but the other ones, they don't have this lock, and the bases seems to stack like on top of each other, which is totally different from SARS-1. So here we only have like this stack between the A1 and A2, but the rest is moved away. And that's caused by this specific promontory region that contains the aspartic and the lysine. So we really wanted to know what it was the role of these residues. If you overlay the structure of these RNAs, you will see how different they are. That like big differences in the cap, but also later after the nucleotide number 2, or you will see stacking in other methyltransferases, but not in SARS. In SARS, it moves away. Then we compared the sequence of different pathogenic coronaviruses from human pathogenic coronaviruses to animal coronaviruses, and we saw that actually these 2 residues are conserved, except for feline, but feline is a different case. We aligned our structures with RNA bound from SARS-CoV-2 with MERS, with only the cap analogs, and we saw that, well, they are in the same place. These residues, aspartic and lysine, this promontory region is there. So this is kind of specific for coronaviruses. Then I just did a structural alignment with the other methyltransferases from vaccinia virus, the human, and the dengue virus. And we observed that here between the beta 1 and alpha A structures, this loop is shorter in all the other methyltransferases, and that we have this 1, 2, 3, 4 kind of insert in this region, as you can see here, that make it like bigger, and that's why we have this promontory region that pushes away the 3rd nucleotide. So this is very specific for coronaviruses, is not present in other methyltransferases, and we thought that this could be actually a very good target for drug development. Then that's why actually we went to the high-throughput facility and start working on understanding what's the role of these residues, and if we do some changes in this region, it could modify or not the activity. So Sarah was really helpful in that. So I talked to Sarah and said, I have this very complicated assay. I have two substrates. It's really hard because our substrate is super expensive. Plus it's RNA, and it will be degraded. So we don't want to take the risk. We need like super clean materials and also robots because we will be testing like for proteins. And, it just takes forever, like 4 hours just to dispense every single thing. And she said, yes, we can help. We have the robots, and that was great. We actually start working together with Shantanu by that time, who just joined the lab. And then we formed a new activity, a new team activity, and we start using robots. And that was very educative, and I really enjoyed working with robots. So here we have Shantanu preparing samples in a biosafety hood that was provided by the facility to prevent RNA degradation. We didn't want to take any risk. And then for protein, we use the mosquito. That is very accurate for dispensing proteins. Then for the substrates, we use the MTase, which is my second favorite robot, but I'll show you later my first or my favorite robot so far. So this is a really good machine that can dispense several times the same reagent. And it was really helpful for our substrates and also our developing reagents after our reaction happened. And they also have all kinds of detectors that you need from luminescence, fluorescence. In this case, we were detecting the luminescence. And we switched from a 96-well format plate to a 348 format. So we could do all this job in a very short time. Well, we could get all this work done and then build these nice plots where we show that changing this aspartic residue by an alanine or a glycine will shift the IC50 to reach the maximum activity of the protein, a little bit in the presence of manganese. But if we delete these two residues, then the activity will go down. Interestingly, here we saw again that manganese favors the reaction. We had some signal detected at very low concentration of the protein, whereas for manganese we didn't saw this increase in the activity, and the shifts in the EC50 of the protein were more pronounced, again showing a high preference for the manganese in this reaction. So that was really good. That's how we finally got published this paper, thanks to all the great job done by the crystallography team and the activity team and the high-throughput facility. And I just want to mention something that I consider very important. So we started with Paul doing one person manual pipetting. Like the maximum that we could do was 30 reactions per day, and it took like 6 hours. And we were using 90-well-6 format plates and like a volume of 50 microliters. From that, I started like trying to improve and get like more tricks in the assay. And I was using multi-channel pipetting. Then we were running like 40 to 45 reactions per day in a 96-well format plate, still 50 microliters volume. But then when we went to the high-throughput facility, we did it in 2 hours. And then we used like 2 or 3 robots, and then we changed to a bigger format so we could run like around 100 reactions per day. And we reduced that 5 times the volume, so that reduced 5 times the cost, which was really good. And that's how we could build like these nice plots and then published our paper in Science. Then later, we really start doing drug discovery. So I joined this project with the Chicago Biomedical Consortium, and we are testing different molecules based on the previous studies and where we find like druggable sites. And this is my favorite machine so far. Because yes, this can go faster, and this is the ECO, and this robot is very nice because it uses ultrasound for dispensing. You don't need pipettes, you don't need tips, you can have accurate dispensing in the nanoliter range. And then we pass from hours to just minutes, literally. It takes like 2 minutes to dispense whatever I put in the source plate. I use mostly this for the compounds because you really want to have a loaded volume. You don't want to waste your reagents. And then you put that here in the source plate, and then the assay plate, And I really want to show this because it freaks me out, but I like to see it all the time. So you put all just your reagents there, it gets in, and then you put your assay plate, it flips over, and then get in, and then the machine will be dispensing. You just create a map, right? Like the well A to the, from the source plate to the well ABC in the assay plate. And that's how you get everything dispensed in just minutes. And then you're really happy because you don't spend like 6 hours just dispensing your reagent, and you can get a ton of work done. So yeah, with this, I just want to highlight all the benefits of working at the HTA facility. It's just like you really save time, effort, and money. And it's really great having this experience personal. Like Sarah was really great helping me, like what machine will be the best for every single step that I have to dispense. And also Michael, who's always there helping me, even like 7 p.m., 8 p.m. It's like, Michael, I don't know what I did. And then they always help us out with all the issues that we have. And so thank you, Sarah. Thanks for all the training and the great job. And also thanks to all the CSGID team. They are hidden somewhere in all this job. I don't present all of them, but they are all important for all these papers that we've been publishing. So if you have any questions, I'll be happy to hear. And I will just pass this movie because I like it. And it shows the reaction.
So thank you. That was great. I don't know. Is Erin?
Erin,
Do we have questions from online users?
I can't see if we have any questions.
did you find any drugs that?
Not yet. Not me. Not this team. But we are working hard on it. Not yet.
You want to tell them that they can submit questions to the chat.
Yeah.
Right now, I don't have any questions thus far.
Okay, so if you want to send any questions in the chat, I don't see anything.
So what's your plans for the drug discovery? Are you going to do targeted, or are you going to do known inhibitors, or are you going to do some screening, or how? What do you guys do?
So far we are doing like screening with some analogs of the substrates. We have some hints that some of them might be working, but yeah, we're trying like to prove that they actually work, because we actually need to set up another kind of assay. We are having a lot of background noise from different compounds that are not 100% compatible with the detection methods. So yeah, that's something that we've been working in the past couple of weeks. Like when we went there, when we switched the assay, it's not working great neither, but we have still another one. And we are also trying to go back to the radioactive method, and we might need to set it up to test all these molecules that are not compatible with the detection methods.
No questions.
Okay.
Well, thank you. That was really great.
Thank you.